All right, so let's get right into it. What we have here is something special. This entire explainer comes from a really unique source, the raw, personal lab notes of a home scientist who's on this quest to master plant tissue culture. It's this amazing story of obsession, of constant failure, and honestly, the pure joy of trying to create something, one little petri dish at a time.
You dig through hundreds of pages of these notes, and you find this one line, isolating meristems again. Now, meristems are basically the stem cells of a plant, right? They're the very source of new life. But the most important word there? It's again. That one word just perfectly sums up this whole cycle of incredibly difficult, ambitious, and, let's be real, super frustrating work.
And that really gets to the heart of what we're looking at today. We're not just talking about putting a cutting in some soil. This is way more fundamental. What does it actually take to grow an entire living plant from just a handful of microscopic cells in a glass jar? Well, let's find out.
So every story needs a beginning. And for our scientists, it all starts with this beautiful, almost magical goal. It's this dream of perfect, sterile, clean creation. So what exactly is plant tissue culture? Simply put, it's a technique where you can grow a whole plant from just a tiny piece of one, like a few cells tiny, on this special nutrient-packed gel. If you get it right, those few cells will multiply and grow into a perfect clone of the parent plant. I mean, just imagine creating a whole army of flawless orchids or monsteras from just one little leaf fragment. The potential is absolutely incredible.
But, and this is a big but, this perfect, sterile dream has a major villain. A powerful, invisible enemy that's lurking around every corner, ready to ruin everything.
As you comb through the lab notes, page after page after page, one word shows up more than any other. It's the scientist's arch nemesis, the ghost in the machine that can destroy months of careful work in just a day or two.
Contamination, that's the one. Bacteria, fungus, mold. These microscopic invaders are literally everywhere. They're in the air we breathe, they're on our skin, on every single surface. And for a DIY biologist trying to grow life on a nutrient-rich gel, well, that gel is basically an all-you-can-eat buffet for these unwanted guests.
But here's what's so amazing. You see this note after another failed experiment. It doesn't just say contaminated, it says very beautiful mold. The entire petri dish is overgrown. There's this dark, almost poetic humor to it. It's the voice of someone who has failed so many times that they've actually started to see the strange beauty in the disaster.
And this table right here, it just shows you the brutal scale of the problem. Experiment number 44, contaminated. Number 76, mold. Number 117, contaminated. We're not talking about a couple of bad days here. The log details a relentless war of attrition against an enemy you can't even see.
So, how do you fight this invisible war? Well, you basically have to become a modern-day alchemist, brewing up these precise elixirs of life not in some ancient dungeon, but right in your kitchen-turned-laboratory. The secret sauce is the growth medium. The recipe is famously known as MS, after its creators, and the logbook details the whole ritual. You have to mix these nutrient powders and vitamins, add some sugar for energy, get the pH exactly right, down to 5.7, and then, the most crucial step, you autoclave it. You cook it under high-pressure steam to kill absolutely everything else in there. But food isn't enough, right? You have to give the cells instructions, and that's where plant hormones come in. It's a delicate balancing act. You've got auxins, which basically tell the cells, hey, grow roots. And you've got cytokinins, which say, okay, time to make some shoots and leaves. Getting that hormonal recipe right is like being the director of life itself. And this brings us to the sheer grinding effort of it all. This is not some casual weekend hobby. These lab notes reveal a relentless, all-consuming cycle of trial, error, and repeat. Just look at this timeline. The journey starts in January 2024. By July of the next year, just a year and a half, the log has passed over 400 separate experimental attempts. Can you imagine? That's not just trial and error. That is a massive, systematic, and frankly grueling campaign.
And a huge part of that campaign was finding the right weapon against contamination. The notes are just full of different sterilization methods. You see tests with regular household bleach, with hydrogen peroxide, with specialized fungicides, even a pro-grade biocide called ppm. Each one is a different battle tactic in this ongoing war.
And then, buried in all these failures, there's this moment of pure clarity. A note that reads, "A failure in the technique, need to clean the air." This is it. This is the scientific method in its purest form. It's not just about doing the same thing over and over. It's about stopping, thinking, and realizing, okay, the problem isn't the gel, it's the air itself. And then, after all the struggle, after hundreds of contaminated jars and probably countless moments of wanting to just give up, the story changes. The persistence finally starts to pay off.
A new phrase starts popping up in the log. It's simple, it's understated, but after everything we've seen, it feels so powerful. Хороший результат. A good result. I mean, those words just stand out like a beacon of hope after a long, long storm. The sterile dream. It's not just a dream anymore.
This chart right here, this puts it all into perspective. On the left, you have this massive bar of over 400 attempts. And then on the right, that tiny little sliver representing the clean, successful cultures. It just shows you the insane odds. Every single one of those successes is a hard-won victory against this overwhelming tide of failure.
And these aren't just abstract successes. They are real, tangible wins that the log celebrates. A cactus developing its first tiny, perfect root in the gel. A calentule plantlet starting to multiply into a little green colony. Even something as simple as a tomato seed sprouting, perfectly clean and sterile. These are the moments that make all that hardship completely worth it. But as we get to the end of this log, you start to realize something really important. For a true DIY biologist like this, success isn't the finish line. It's just the starting pistol for the next race.
The last numbered experiment in this particular log is 414. But the notes, they don't stop there. The work just keeps going. Success isn't a reason to sit back and relax. It's the fuel that makes you want to do even more.
And the to-do list at the end says it all. The ambitions have only gotten bigger. There's a whole new project plan for orchids, a note to make a how-to video for monsteras, another to test new hormones, and then, right there at the end, that phrase that started it all, isolate meristems again. The cycle continues.
So it leaves us with this one big final thought. This whole journey was this epic battle for a perfectly sterile environment. But when you look back at the hundreds of notes on failures and adaptations and those small precious victories, you have to wonder, was perfect sterility really the point? Or was the process itself, the trying, the failing, the learning, and finally the succeeding, the real discovery all along?